understand it. No problem.
I guess I'll let go of it there. Okay, we have everything but the part we want in the box.
Do you ever use both of the arms in tandem to like push and move? Or is that impossible or challenging?
I haven't really seen it done too much with samples, but with instruments and other engineering related dives, we'll bring out the other arm. Hold on to a giant piece of subsea equipment and push in connectors with the craft arm.
I think I'm going to cut it here, and then that way you'll for sure get the branch.
That'll work.
Well,
Can we get some pictures of where that's cutting?
Yes.
Not for sure, but maybe likely.
Mm-hmm. Nice.
You.
I'm thinking of a Queen song right now.
Yeah. We have some viewers saying that Dan must clean up at arcades with the claw arcade game.
Actually, I'm horrible at that game. My 10-year-old's better than I am.
I heard they make them really, like, statistically not, you don't win. Statistically, just doesn't work.
Well, we might cut it again, because it's kind of like trying to scratch your right armpit with your right arm there. The box will close there.
Yeah, we could just do that, I think, yeah.
Whew. Do you want me to close it all the way or leave?
Yeah.
Okay.
I don't think they're bothered about that piece. It'll kind of wedge in there.
Yeah. There we go.
May even break off.
Yeah. Want me to bump it out?
Nope.
Okay.
Let's see.
What was the other one you were looking at? The sponge?
The sponge. I don't think I want to collect that.
You wanted
room for it.
That was the one you wanted a side view of?
Yeah, the side view is great.
Okay, video, you can zoom in there. Ah, sorry. My bad.
Hi, Magda.
Okay, I think let's go ahead and fire a Niskin here. I know we haven't moved a lot in depth, but we've got coral around, and we could get some eDNA.
Do you want full wide again?
Yeah, we'll need the big camera for that. You guys have seen enough of the sponge, have you?
Yeah, we got the sponge. Saturated on the sponge. Just kidding.
And just so you know, that previous sample was sample 53, or 52, and now we're on sample 53.
Thank you.
Thanks,
You
Taylor.
want to hit the porch
Yep,
light?
no problem.
It should still be on. Yeah.
Really?
You know, we
Oh, it's
cycle it? Yeah.
iris.
All right. Now it's off. Now it's on.
We've taken no Niskins yet, is that right?
No, we've taken one.
Yeah,
So
7's
7
tripped,
was tripped, yeah.
8's disabled, 9
Oh,
is
8's
next.
disabled?
8 is
Yeah.
disabled.
Oh, I didn't know that. So we only have
9
4
it is.
Niskin bottles.
We're taking number 9.
It went.
Yes.
Just tripped, didn't it?
It did,
7,
yeah.
8, 9. Yeah.
Yep. So you see,
Thanks.
And Danny, can you remind us what these water samples are analyzed for? The water samples are analyzed for 2 things. One is eDNA, which is sampling DNA in the water column. There's this cool sea spider at the bottom of that frame we could zoom in on. eDNA is taking water samples and cells of animals that have, so animals slough off their cells, and you can collect water samples and then analyze it, the DNA that's in there, to see what animals are around. And it's also analyzed for carbonate chemistry of the water. And so throughout our expedition, not only are we live streaming on nautiluslive.org, but we also do something called ship-to-shore interactions. So schools and museums are signing up to talk to scientists on the vessel, and we share with them some of the cool findings. And it's really cute because Erin, the video engineer with us right now, she did a couple of these, and one today, and that family and student are tuning in right now, so they're really excited to see Erin doing her work in real time.
Hi guys, thanks for tuning in.
And of course they're curious to learn more about the cameras whenever you're ready.
So the main camera screen you're watching in feed 1 is by a company called Insight Pacific, and it's called a Zeus Plus. So it's an HD camera, and it's in titanium housing so that it can go really, really deep.
Amazing. New fact for me, titanium.
Mm-hmm. And it weighs, when it's in the air, it actually weighs close to 73 pounds.
Jeez. And then we know that this area is pitch dark and we're lighting it with all of these, you know, bright lights. So how do you find the right adjustments to most credibly represent the colors we're seeing? Can we get a zoom on that stalked animal?
Yeah,
That's
you can
upper
zoom in
left.
there.
Yep.
A different kind of stalked crinoid.
I have a little bit more when we get closer.
I think it might be a stalked crinoid, but it's different than the ones we usually see, which are usually yellow. That's cool. Okay, we can zoom out. So
Good to go.
we want to keep moving to the
east up the canyon wall, or along the canyon wall, I guess, and moving towards our the 38.38, I think.
There's a great view of Hercules from
Moving
Argus
east.
right now,
What
showing
was
the
that?
steepness of this habitat.
Aaron, are we full wide on Argus? Nope. Okay.
I'm just pushing in past the corners. Now we are, with just the corners gone.
Nice. Yeah, I was just curious.
That is a great shot. Can we get
Yeah.
a picture of that too?
I already got it.
Thanks, Dorian.
Yeah, that looks amazing.
I had a publication last year contact me. They wanted pictures of the steep walls, so that would be a good one.
Sorry, what was your question earlier?
My question was, how do you make adjustments to most accurately capture the colors that we're seeing in such a deep environment?
So it's a combination of trying not to blow out the highlights, so the whites, and also trying to keep it lit enough so that the pilots, I mean, they're the priority, so you want them to be able to see where they're going. So it's finding that balance. But some of these sponges are very, very white and reflect the light quite a bit.
The sponge that just went off the top of the screen there was really neat. It's like a face sponge, but I think it's on a stalk again. These stalk sponges are really neat.
And evolutionarily
Push
speaking,
in a bit there if you want, Aaron.
what are some advantages to being just a stalk versus having direct connection or, you
Okay.
know, to the rock?
Yeah, I don't really know too much about these organisms. We're seeing them for the first time, but I imagine it has to do with getting them out to the water column where they can access food versus being more securely on the rocks. I don't know.
Neat, yeah.
Yeah.
Seems like we're seeing longer tentacles on anemones and these stalks. I mean, it kind of seems like that could be a reason why.
Yeah.
Do you want wide again?
We can go wide.
You good with that, Herc?
Contact.
What's that orange floaty blob? Like some kind of jelly? Can you zoom in on that?
I can zoom in, laser zoom if you want. Perfect, thank you.
What is that?
Who are you?
Oh, look, it's attached.
Yeah, I thought so too, but
I think we've seen something like this before, but I can't remember what it was. It was kind of bobbing along the bottom before on one of our previous expeditions. But this one's kind of attached to the
Push in a
side.
bit more if you want, Aaron.
Can we zoom more, or is that it?
That's all I've got.
Thank you. This could be our purple orb. Except it's more like a firework.
Yeah.
Whoa.
It's got a lot of color.
That's cool. I don't know what that is.
What's that?
I said, that's cool. I don't know what it is.
I don't even know what family that might be in. Is that an Idarian? Seems like it has tentacles of some sort.
Yeah.
Yeah, it seems as if though, like all the creatures down here are stalked or have longer tentacles to help them maybe feed more in the
Seems
water
like
column.
there's a really long tentacle holding on.
Ooh.
To the base.
Yeah. Very cool.
Okay, we can go wide.
Moving on.
We have an orange orb of some sort.
So Jenny, you pointed out these steep slopes on this feature that we're seeing. And on land, often steep slopes face issues like erosion. Deep in the ocean, are there any physical, I guess, changes that these deep slopes go through similar to erosion or erosion itself?
Well, I think technically, as a canyon, it is to move sediment through from top to bottom. I don't really know that much about geology of submarine canyons, but they do erode over time just from being here, and I think sediment runs through the canyon as well. Danny, do you know much more about that? And that's kind of one of the functions of a canyon, right? To move sediment? Not really function, but result of. It's like a slide.
Yeah, yeah. So, I mean, if you think about a canyon on land and the water forms it and then water moving through it. So that's what happens with sediment too. Well,
There are underwater landslides. Yes.
Guy. Underwater
get a
landslides.
zoom on that sponge. It looks like kind of like the one we collected, and look, it might have a long stalk on it. Get it out from under that ledge.
Go ahead, Eric.
Wow.
And that big red shrimp. Cool.
And some of our listeners were saying that potentially that species was a dandelion ctenophore, which Nautilus has seen on other expeditions.
I knew somebody would write in and tell us.
Awesome.
Thanks, people tuning in. Helpful.
We can go wide.
Was there a question for me?
You answered it with the landslide tidbit. Thank you.
Short and sweet.
This guy's specialty.
So we not only have our control room with a number of scientists and specialists, but we have scientists on the vessel that often listen into our dives, and they're on their own watch, but they are here to tune in when needed to provide their expertise. So Guy is a geologist and works with USGS and often answers our questions regarding the rocks and the features that we're seeing, the more sedimentary features. But then we also have this capacity called telepresence. So in real time, we're sharing our video and data with scientists, and that's really wonderful because experts on land can also contribute to
Below those
what we're seeing.
colorful anemones, I see like a split stalk, kind of the same color as the rock background. Could we take a look at that?
Gotcha. Should be good for a laser zoom.
It's like a dead something, maybe dead, one of those dead sponge stalks or something like that. With some stuff growing on it. Zoanthids maybe. Okay, thank you.
Dandelion ctenophore, pretty cool. I'm just looking up some pictures and it seems like it's something that has a lot of tentacles out, attached to the bottom.
I'm gonna try and scoot under you there.
To our ROV
Oh, look at
pilots,
that big sponge.
yeah, is it harder to maneuver the ROVs in a steep sloping environment or an environment that's flat but has a lot of rocks, big rocks or boulders around?
I guess that would depend, but for me it's easier to come up a cliff face because at that point we usually take one of the dynamics out, and that's moving the boat. So when we're moving the ship sideways along the cliff face with another cliff face in the neighborhood, it's
There's something swimming on the right side of the screen there. What is that?
Pelagic cucumber.
Ooh. Oh, just got
I
knocked
like
off.
the way you move.
Oh, pelagic cucumber. Oh, wow.
can zoom in a bit on him if you want. So.
Ooh.
It sure looks like a,
It's like a nudibranch.
Look at the guts. Cool.
Aw.
That's amazing.
Wow.
See all of its
Yeah,
internal
she's really
structures.
good.
Holy moly.
That is really cool. Aw. Another Halloween costume.
Yeah, literally doing the worm in the air. Yeah.
Oh my gosh.
Look
that
out
is
for that
so
camera.
cool. That camera is amazing.
It's too close.
Oh, it's
Can't
so
focus
cool.
anymore.
Nicely done, Eric.
So is that different than the sea pigs we've been seeing? Did it just get knocked
Ooh,
off and is
is that a
swimming,
crab?
or is that?
Don't know.
Yeah.
It's a crab
Don't
coming
see
up.
any around here.
Maybe.
Looks a little steep where we're at.
That's a big squatty.
Ooh.
Wow.
What is that? Squat lobster?
Yeah, big squat lobster.
Zoom in on him a bit if you want. Or her.
There's a coral right next to it. Or zoanthids.
Yeah, again, that looks like a coral axis with some zoanthids on it, similar to what we saw before.
Sometimes these rock features just have so many different animals on it and really speak to the habitat that these corals and sponges create and find on just a, single rock.
Okay, we got a great look at that. Thank you.
Can't look. It's disgusting.
Move along, move along.
Grossing me right out. Sorry, I didn't know the part.
When we were panning over, I think we saw another spoonworm, but I'm not too familiar with that animal. Can you tell us a little bit more about spoonworms?
Oh, I have to look that up. I don't know too much about them myself. They lay out on the sediment like that, and then when they're disturbed or when they capture food, they pull back into the little burrow, but that's kind of all I know. All right, I'm happy with that. Thank you.
I hope so, because I'm going to have Predator movie nightmares tonight.
It's just a crab.
Just a crab.
What have we got to the left of that anemone? The big white anemone that's on the upper left corner of the screen.
Oh, yeah, I see it. Is that better with that porch light off?
Yeah, it's a bit better.
Can we get a zoom on that coral that's to the left of the anemone?
Yeah, you can zoom in there. I'm actually touching the cliff there, so.
Maybe that same Narella that we were looking at before. I think it probably is.
Happy?
Yep, that looks good.
Are there more softer corals in the deep ocean than in our shallower waters?
I think there probably are. I'm more familiar with the deeper stuff than I am with the shallow ones, but I think there are more soft corals in the deep habitat. That's a great question for Gary Williams.
Guy, are you still in here? Are still listening?
I'm listening.
Is that geology we're looking at there?
Well, the black is oxidized rock, yeah.
That's it? That's all I get? Oxidized rock?
Antonella, I was talking a little bit about ROV adaptations with Paul, and he was telling me a little bit about how static is being used in space, and we know how we have the slurp, so kind of like a vacuum, it's used underwater. But what are other adaptations in the future for ROVs that will help with deep ocean research?
That's a good question. I think that could take a lot of different forms. I mean, I personally work on autonomy, so I think my answer will be a little bit biased, but I think of one example of my first cruise last year. We were looking for hydrothermal vents, which involved like looking for water shimmering in the camera, which sometimes was really hard to find. And I could imagine like having some level of like automated help for that. Like the geologists, of course, were really good at it, but there were that could be a potential area for like computer vision to be sort of as an assistant detecting areas of interest. Some of the stuff I work on, not in deep water, but involves like online automated detection of coral species, which could potentially help if you can get at least rough estimates of what you're seeing. And the sort of biodiversity in an area.
Very cool. These really tall black corals, pathies, they're so impressive. I haven't seen them quite this large.
It's so cool to look at Argus Cam just to see what we're doing here. Very vertical, just Hercules totally holding steady while we look at these walls. It's just so neat to think of it three-dimensionally, what we're seeing.
That is a neat view, and it's neat to see the Herc just scooting along the wall sideways.
Is that sponge stalked as well?
Yes, it is. And
Yeah.
there's one of our
another
dandelion
dandelion
ctenophore.
Thanks to our Nautilus viewer for helping us out with that one.
And it seems like viewers are getting super interested in deep corals.
Let's take a look at that corkscrew type coral, spiral coral.
Copy.
Do you have any resources, books, or online
Aaron,
resources?
You're gonna look over there now, Aaron. You can see.
Well, NOAA has a deep sea coral program that has a lot of great information on the web about it. NOAA's doing a lot of work to map and identify where corals are. Partially.
This might be Radicipes, which we collected in 2017 and was a range extension from the Aleutian Islands.
Ooh.
And it's distinguished by the polyps all being on one side of the axis, which it looked to me like that was.
So I would look at the NOAA Deep Sea Coral Program as a great resource for learning more about deep sea coral and NOAA's role in helping to learn more about them and map them and conserve them. And we have an education resource through the National Marine Sanctuaries that is about West Coast National Marine Sanctuaries and deep sea communities, the deep sea community curriculum. And again, you can just Google that. Deep sea coral communities or deep sea coral curriculum sanctuaries.
That's all the Zoom I have for you scientists.
Okay.
And students get to do a little transect
That
of
looks
deep
good.
sea habitat.
We got a lot of those collected last time, so we definitely don't need to collect it, but it's I got a good look at it. Thanks.
Happy for me to zoom out?
Yes.
We're gonna dust it a bit. I drifted into the cliff there and just need to back up a bit before we move on.
Earlier, Danny and I did an interaction, and person in the on land said we looked like we were astronauts going to outer space. I feel like we kind of are, but on our own planet, discovering things right here on our own planet.
The last time we were out on Nautilus, we were really focused on some of the corals, and we did get some sponge samples, but not as many as the coral samples that we got. And when we realized that there was so much we needed to learn about the sponges, so we've been collecting more sponges this time to learn more about them. They're really tricky because they have so much varied morphology, so it's hard to, we're still learning a lot about their taxonomy. So that's why we've been collecting some sponges during this dive and on our other dives.
There's quite a bit of work being done about the use of marine sponges in terms of medical applications. And I didn't read too extensively about it, but it seems like there's some properties in sponges that can slow or stop the growth of cells that could be anti-cancer treatments. So it'd be really interesting to learn more about the benefits of sponges for so many applications.
And I feel like there's a fine line between an animal's benefit to humanity. There's interesting relationships between us sometimes realizing those benefits and having tighter regulations around harvesting of them and or use of them, but then also there's more of a target on that species. So there's really
one of those swimming
pros and
sea
cons.
cucumbers.
Well, we have a prohibition of take of invertebrates in our sanctuary. So we have a permit to collect on this expedition. So we can learn the names, learn these species and be able to properly identify them.
And I know one thing, they could, if this is something that's in particular interest for the medical field, they could study like the way that it actually functions. So like bats, they actually found like anticoagulants because they studied like bats and how bats can suck blood from other organisms and kind of get rid of that like clotting ability. So that's something that they've studied in bats.
You're muted.
Can we take a look at that sponge? It's a, that might be a white
I read
Picasso
something similar
sponge.
about those leeches we were seeing back on those fish, that they have an anticoagulation ability when they get into the fish.
Did you say this is the Picasso sponge?
I thought it might be. I'm wondering if when we slide to the side, if it's
Push
going to
in
be on
there
a
if you want.
stalk. It looks a little different, actually.
Seems like the holes are bigger.
Yeah.
We have an interpretive dance for Picasso sponge.
We did. I got to see it.
Oh.
So this is like dream come true to finally see the Picasso sponge if this is what it is. Since I know the dance now.
Oh, okay.
Floppy.
So this might, Caitlin tells me this looks like one we collected in 2017. If we see that it's on a stalk, one of the Calophocus.
So before we go on any of these expeditions, we map the seafloor. So then we can highlight certain features, understand how to best navigate our ROVs.
That's
But
great. Thanks. Could we come zoom out a little bit, and then as we slide around it, could we look back and see if it's on a stalk? Sorry, Carrie.
no
Copy
problem.
that.
Do you want me to zoom out a bit more? Or are you happy with this?
I think that'll work. I think it'll work here.
I'm just going to back away before I come around there. Well, I'll try.
But earlier Jenny mentioned that going this deep in the ocean is, you know, almost comparable to going to space, just because no one's ever been here and there's still so much to learn. And so I was curious if Miles could tell us a little bit about the initiative, the Seabed 2030 Map the Gaps initiative, and different ways of mapping, different resolutions of mapping our seafloor.
The JEBCO Seabed 2030 initiative is really ambitious in its scope. I think it is what is needed at this point. I don't want to be pessimistic and say we won't get there, but it's really ambitious to sort of try and map the entire ocean floor to a resolution of 400 meters or better. But with that said, it is sort of unifying the effort to get there. So in the past, most UNOLS vessels and research vessels will turn on their multi-beam whenever we leave and return port, and you get these cow tracks, just areas that have been mapped repeatedly over and over and over. So the Seabed 2030 initiative is sort of pointing this out and saying, hey, like, why don't we, add an hour onto our transit time to just step over one swath width and map a new area, and sort of they're making this big push to map the gaps. And so all these different sort of cow tracks have been laid down, and now this is sort of pushing people to sort of aim for the areas that have yet to be mapped. Even if you cross over a lot of previously mapped areas, you're hitting sort of little donut holes of unmapped areas.
Yeah, this one has a stalk too. Boy, this is really a beautiful one.
Miles, are there autonomous underwater vehicles that can map?
There are. HUI and a few other institutes have AUVs that have side scan and multi-beam on them.
We can move on. Thank you.
There are some oil companies that have entire fleets of autonomous vehicles that they'll, throw 5 or 10 of these down and map large areas at very high resolution. But that sort of funding and money is not always available to research scientists.
Would that data not be available to other scientists, or do you think it's proprietary?
A lot of it is proprietary. When I was in my last job working on sort of the effects of deep water corals from the BP spill, we got access to a lot of BP data, and it was amazing the resources they had, how quickly they could acquire data, and the sheer scale that they were able to acquire data.
Interesting.
Can we zoom in on that kind of translucent
Organism?
yeah, thingy?
You can zoom there, Ashley. Aaron, sorry.
Wow. Okay, I think this is one we haven't gotten yet, right? Okay.
No.
That's what I was thinking. Okay. We want to collect this. we can do it in a slurp.
What is it?
It is, we think it's let me say it right, umbellula.
Interesting.
Umbellula. I always say that wrong. Umbellula.
Can you spell that?
U-M-B-E-L-L. Umbel, and then U-L-A.
That's beautiful.
Yeah.
Good zoom.
We've been keeping an eye out for this. So this is 3311 meters.
Gonna be a bump here.
Huh. What else do you think it could be? It's still interesting, huh? Maybe we should just get it.
That is really cool looking. Oh, look at the amphipods on there too.
Yeah, I don't know. What is this? Did I hear Jan say echinoderm?
Ah, really?
Is that like a little crab on there? On the pink part?
Yeah. Or maybe a sea spider or something?
That is beautiful.
So it's maybe not what
Ooh.
we thought it was at first, but still don't know what it is, so we can go ahead and
Viewers are
slap it.
suggesting that it's a solitary hydroid.
Oh. Interesting.
We got some smart ones on tonight.
I know.
Thank
Thanks for your
you.
help, listeners. We're seeing things for the first time.
It takes a village.
That's what I love about this.
I don't know. What do you guys think down the lounge? Solitary hydroid.
Sure looks like it,
Hmm.
based on my quick search here.
So in the control room, very few people have access to internet. So most of what we're saying is just from our previous work and knowledge.
This looks like a, I don't know if it's the same species, but certainly looks the same body type.
Is that
That's
a
super cool.
picnogonid exoskeleton in there?
I think there's two. There's one at the pink part and one up in the tentacles. Here's a question. Do we need a sample of this? Since it's likely not the umbellula.
Yeah. All right, we got some great images of this, but we determined it's not what we were looking for. And thank you to our listeners or viewers helping us figure out what this is.
Super
So we're
cool.
gonna, I don't need to collect this, so thank you for stopping and taking a good look. Oh gosh, you guys. All right, I'm getting some more input from our viewers in the lounge, and we do want to get this. If we're still in position, can we go ahead and get this?
Copy.
Ship is ready.
Are there any rules or practices in national marine sanctuaries around collecting the first example of something you see?
There are not any rules, but we do use some judgment and common practices around that. So before the expedition, we put some time and thought into evaluating what specimens, what species we're still want to study and want to collect, and we need to collect. We haven't collected it before. We still have questions about, and we have a long list that we make a little bit shorter and then prioritize those things. So usually we like to see a few examples of the specimen before we collect it, but there's also a risk that if we pass something by, we won't get to collect it again. And if we think that it is going to be a somewhat common species, we just maybe haven't been in that habitat yet, then we would go ahead and collect it. So we just evaluate how much of the area we're serving and how much we have time for and how much space we have time for and what our priorities are.
What jar do you want this sample in?
So we have slurp jars 4, 5, 6, and 7 available, so any of those are fine.
Okay.
Are we still slurping, correct?
4 will work.
Yeah, that's how you want to get it, is it? Looks pretty soft.
Yeah, I think it can be slurped. I think it's slurpable.
We'll go for a slurp.
Okay, so which jar are you going to go for?
We're going for jar 4.
4, okay.
Really?
Taylor Ann, can you tell us a little bit about when we use the slurp?
Yeah, so we use the slurp when we think that we can collect like the whole species of an organism, the whole body, without disturbing or ruining its structure. So this guy looks pretty easy, like it can just get sucked up in one whole piece. It'd be a little bit easier than trying to use the manipulator arm to grab onto it, which could probably crush it because it is soft. So that's why the slurp would be the best bet. But you have to be careful as well. Sometimes the slurp is a little bit too strong. But I think that'll be a good choice for this sample.
Can you hear me?
I can hear you, Dan.
So we'll probably go to, I don't know, what do you think, Mouse? 30%?
That'd be a good place to start.
Okay.
The last watch recommended flushing for a little bit.
Solitary hydroid made the CBS
So are you at
results
30% now?
We are.
some
Yeah,
years
so
ago.
it's flushing.
No, we already flushed it.
Roger.
Yeah, we flushed the flush jar and then we flushed this jar a little bit.
Gotcha.
So. Aaron, can you zoom in a bit there for us? Thanks. Yeah, maybe a bit more. Perfect right there, thank you. So now we, as soon as you see it comes into the jar, you can kill the
Okay.
kill the suction
Okay.
if it comes
There it
into the
comes.
jar.
Something's
It's
coming.
coming. There's a cloud.
Are we on 053 or
Should I kill it
054?
now? Oh,
There it is.
That was
you can turn
054.
off suction.
054.
it's off.
And then
053
you can
was the,
go the other way
skin
on the suction jar
sample.
back to the flush jar.
Okay.
Aaron, if you zoom out for me, I'll
Yep.
put this hose away. Thank you.
Okay.
Yeah. I got it.
Great.
Thank you. Thank you very much.
Can we control the slurp pressure that it pulls at?
While we're here, can we do a zoom right where we are on there's kind of like some stick animals, something like sea whips or something on the wall, or maybe they're stalks of old sponges just below the lasers, below the anemone. One's kind of translucent and one's kind of mud colored. Below below the pink anemone.
I can't hear her. I can't hear her over there.
Sorry.
That's better.
Is that better? Okay.
Yeah. Were you
Sorry.
asking a question of us?
Could we do a zoom right here, right where the lasers are actually now? There's kind of like some sea whips or something. One is kind of translucent and one is like mud colored. They're kind of blending into the wall now. I can't really see them, but right where the lasers are.
No, I'm coming around to take out the wrap.
I backed up.
Tell me when you're ready for a zoom.
Yeah, ready for zoom?
Okay.
Sorry, where am I looking?
That's
Up,
okay. You
down,
can
left,
zoom out a little bit, and then I lost one of them, but one of them's still to the right, and it's kind of translucent. Oh, now it's kind of pinkish off to the right side, upper right corner.
Ah, I see, I see.
See there?
Yeah. This guy.
Yeah. That looks like a black coral, maybe?
Sorry, I'm touching the cliff.
I think it looks like the one you guys collected earlier. Okay. All right, we already collected one of these, so we can move along.
Roger, moving along.
We do have a few questions about our slurp capacity, slurp feature. So one was if we could control the pressure of the vacuum, or...
I can barely hear you over there.
Oh, we had a question about our slurp feature. One was, can we control the pressure that a specimen is slurped at? And then two, they were curious, how can you tell the specimen gets to the jar?
You want to take that one?
Sure. Yeah, so we can control the valve and open it a certain amount from 0 to 100%. So 0 is off and 100 is full suction. So we did that one at 30% because it was soft. We didn't want to damage it. And then we have a utility cam that we're looking at on the big screen above us, where we see all the jars rotating around, so we can see what jar it's going into, and then we can watch it. And then when the specimen comes into the jar, we know we've collected the sample.
Oh, nice.
Pardon my dust, remodeling the cliff a bit here.
Looks like Aaron added that one shot onto the quad camera, quad view, excuse me.
Yeah.
So that's the camera the ROV pilots are looking at to see if the specimen is collected.
Oh, wow.
I believe that's where we were thrusting parked while we were parked there. So slide up current there a little.